Um, today is the 8th, and so we'll be looking at cell membranes and passive transport. We'll possibly get into active transport a little bit too. And so make sure that's unlocked. Probably if you're like the type of student that whenever you take notes, like if I were drawing something or writing something, you tend to like put whatever I write down. Then basically for this class and like next class, I sort of explain everything on this one giant cell. So if you tend to like draw the pictures that I do or whatever, I take like just a whole page, leave some room on the edge, because you have to like have stuff you can write on the outside, but I just draw like a big old like oval on like an entire page. Because like I said, I'll probably teach a class and a half with just this one cell. I'll still show pictures and things over there, but it'll just be kind of like one big cell. Hayden, cool. And then along the way, if you got questions, let me know. So how are you guys doing? Good. Okay. Last time you just kind of like, all right. Okay. So last class we were looking at phospholipids, and if I dump them on water, they automatically made bilayers. And then if I take that phospholipid bilayer and I put it underwater, what did it automatically do? Yeah, like form a sphere, which is the beginnings of a cell. And then the thing that I was so fascinated about was that when I look at phospholipid bilayers, like these phospholipids aren't bonded with each other. So phospholipids aren't bonded with their neighbors. Like that one's not bonded with its neighbor, bonded with its neighbor that way. They're also not bonded with the phospholipids across the aisle. And so somewhere we left off around the idea that they're like constantly like sliding around and moving around this way, right? Does that sound familiar or not? Okay. They quietly shook their heads, yes. Okay. So if you think of phospholipid bilayer, it's sort of like this. If these are the phosphate heads right here, then they kind of are just shuffling around. There we go. So they're kind of just shuffling around like this all the time. And did I show you that experiment where they like figured out how often they were shuffling around? Okay, so whenever we said frequently, turned out that that was millions of times, 10 million times a second. And then for a flip-flop, that was really rare. And was there anything else that happened with flip-flops? Somewhere right around here is going to be where it's like, all right, we didn't learn that yet. So they don't flip-flop very often. Why was it that they don't flip-flop very often? Why is that not very likely to happen right there? Yeah. Yeah. Along the way, that means these hydrophobic tails have to touch water. These hydrophilic heads have to be basically touching the lipid tail. So this doesn't happen very often. And so if you looked at the picture that I had up here last time, it would have something like this. I'd have a bunch of phospholipids like that. In reality, every other tail would have a bend in it. They're unsaturated. And maybe here's the other half like this. And maybe like this is the cell. So one of the things that I may or may not have gotten to was these can flip-flop. Did I tell you any enzymes that help them flip-flop? Yeah, flipase and flopase. And why would I ever do that? Why would I move a purple one to the blue side or a blue one to the purple side? Why did I take the purple ones and put them on the outside? Yeah, that's a way that I can warn my immune system that I've got a problem. So if I'm a cell, I pick up mutations really, really fast. In Unit 4, we study cell cycle and cancer and stuff like that. And in Unit 4, I'll kind of show you the math. I'll show you my sources, because at the moment you might not believe me. But it's determined that as fast, if all of us were snapping our fingers, like both hands, all of us together, 24/7, as fast as we could, just in my body, I'm picking up genetic mutations faster than that. Genetic mutations can lead to cancer. You're like, well, how do you not have cancer within like the first 40 seconds of life? Exactly. My body is finding it, fixing it, and correcting it. But if we're all doing this, like this is one mutation, 2, 3, 4, 5, 6, 7, like that, plus all of you guys doing it at the same time, again, I promise this is true, I'm picking up mutations in my body faster than that. So my body catches most of them, fixes most of them. But if I live long enough, 50, 60, 70, 80 years, some of those mutations slip by without getting corrected, and so that's why it's common for us to eventually get cancer. Outside of like leukemia, at your age, very few people get cancer. You know, in a school of 3,300 or whatever, it's unlikely. But if I went to like a center, like a geriatric center or whatever, where I've got a bunch of people that are in their 80s, like a good chunk of them get cancer. What's the likelihood that you get cancer in your lifetime? It's worse than that. It's about 40-45% chance. So 40% of us will get cancer in our lifetime. It's worse for guys than girls, which is surprising because females live about 5 years longer, and one of the best predictors of whether or not you're going to get cancer is how long you've been around, because then there's like more mutations that could have slipped through, right? So the longer I live, the more likely I am. So it's kind of surprising that like that happens. So one of the things that can happen is if I've got a cell and I'm sick, maybe I somehow know that I have cancer, and you're like, well, how would a cell know that it has cancer? How would it know to flip this around? That's unit 4. So right now you just kind of have to trust me. But my cell, one of the things it can do is it can take some of those inner leaflet, in this diagram, purple, some of those purple phospholipids, flip them to the outside. That tells my white blood cells something's wrong, and the white blood cell gives me permission to kill myself. Not me as a person, me as an individual cell. So don't mistake that. So this is my white blood cells telling this individual cell kind of goes like this. It's like, hey, I'm sick. And the white blood cell says, yes, you are. You have my permission to kill yourself. And then it does. And that process we'll learn is called apoptosis. Apoptosis is programmed cell death. All living things have a genetic code for cell suicide, and it's conserved. So like the apoptosis sequence in a fly, if you splice it out of a fly and put it in me, it works because it's identical. The proteins that turn it on in me would turn it on in a fly because they're identical. It's just like back in the days of CDs, if you've got your, whatever your favorite band is, and it's like album number 4, and somebody else has it, and it's album number 4, like they're the same songs because they're identical to each other. They're the same digital copies. So it's kind of amazing. A cell can tell on itself by like flipping these things around. So I find that pretty cool. All right. One last thing, just so you can appreciate the stuff that we study in here. You study cells in 9th grade. Then you come back and you study them again here. And eventually you're like, I feel like I know everything about cells. But that might not be true. So let me show you an example. This is a book called The Cell. We're doing a unit on cells, but you can do an entire class on cells. Like that book looks pretty big. This isn't all of it. When books get big enough, they get too expensive to print. So they print some of it, and then they just give you a disc with the rest of it on there. So I went ahead and printed that. So this is the rest of it. What's that? Cells. You're like, isn't Unit 2 in AP Bio cells? Yeah. Is there more to know? Yeah. There's this much. We'll later on discuss the nervous system. There's a book on the nervous system. You're like, wow, is there more stuff to learn on the nervous system? You can major in the nervous system. You can be a cell biologist. You can get a master's degree, a PhD in cell biology. So what we're currently doing as a unit, that can be your entire career. So there's tons of stuff to know. So there's all kinds of interesting things that you can learn along the way. super weird when you hear somebody like behind you rooting around. All right. Okay, so let's say that I'm looking at a liver cell versus a lung cell versus a brain cell versus a kidney cell. Is it possible that I need the membranes to be a little bit different on liver versus lung versus brain? You probably haven't handled a whole lot of livers, lungs, and brains. Maybe liver. Maybe you got like chicken liver and went fishing with it or something like that. But you probably haven't held a whole lot of brains. If so, where? But you might dissect, like, did anatomy dissect sheep brains maybe last year? Yeah. So I used to teach anatomy and physiology like 20 years ago, and we would dissect like sheep brains and hearts and cats and stuff. But anyway, even if I don't know a ton of stuff about those, like liver cells should probably be different from brain cells and lung cells. So sometimes I need the membranes to be like extra fluid or more solid. So what is changing here to make this one more fluid? Like what's causing those bends? Yeah, I'm putting in double bonds. I'm making them unsaturated, and so now I can make them more liquidy. If I, for whatever reason, need the cell membrane to like firm up a bit, then I start taking out some of the double bonds and putting in singles. One thing that a lot of students don't appreciate in ninth grade is like, we always kind of compare as teachers, we compare like the cell membrane to sort of being like the walls of this room. But they're different because the walls of this room we build and then we like leave them. Cell membranes, your body is always tweaking them. So even right now, my body is like taking out some phospholipids and putting in some slightly different ones. It's always tweaking stuff along the way. So it's a very active process. So if I want to change the fluidity, I can just unsaturate, or if I want to change it and make it more solid, I can start saturating the tails more. Cholesterol. So cholesterol is going to start popping up in like pretty much all the units. So starting in this unit, you'll still see it a few more times, but starting in this unit, that's when you want to be able to recognize cholesterol. You don't have to be able to draw it cold, but if you're going to be a science person in the future, you want to get the major shape of it cold, meaning like if I gave you a blank sheet of paper. So with cholesterol, I'll kind of put it in the middle of my cell here for a second, then I'm going to erase it. What I know is hexagon, 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 pentagon. I don't know any of the rest of it. I just know if I have a hex, 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 pentagon, and it's kind of going up almost like a stair step, I kind of picture it going like that, then that's cholesterol. So cholesterol is interesting because we'll look at this several times, but there's all kinds of like hormones that come from cholesterol. And we call hormones that come from cholesterol steroid hormones. Cholesterol, steroid, aldosterone, estra, dialprogesterone, testosterone. So you get a whole lot of ostro and estro and astro. Those are steroid hormones. They come from cholesterol. And as you can see, hex, 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 pentagon, hex, 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 pentagon in a stair-step formation. They don't always have ostro or estro in the name like cortisol. That's the stress drug. But a lot of times they do. So we'll come back to cholesterol a little bit later, but it's got another function. Cholesterol also can be put in my cell membranes to control fluidity. So my body can add cholesterol and take it away, and I'm kind of tweaking the fluidity of the membrane. For AP Bio, that's all you have to know. If you just know, hey, I can add and remove cholesterol and that'll change fluidity, you're good. You're like, all right, well, does adding it make it more fluid or less fluid? It depends on the temperature. So this next part you don't have to know for AP Bio. It's only mainly if you're going to be a biology major of some kind or go into a medical career, whatever. So cholesterol basically does the opposite of temperature. So as I heat up cell membranes, when I heat up cell membranes, should they be moving faster or slower if I heat them up? Faster. If I add cholesterol, it does the opposite. So what would cholesterol do then? Slow it down. When I start cooling membranes, they start moving slower. So if I add cholesterol, the opposite of that would be they stay more fluid. They kind of stay moving faster. Yeah. So as far as how that works, the chemistry on it's kind of weird. So I just avoid it entirely. In AP Bio, again, I can know if it's like, hey, how can you change fluidity? Well, more double bonds will be more fluid. More single bonds will be more solid. And I can start adding and removing cholesterol, and that can tweak fluidity as well. Again, I don't have to know what happens when I add more, what happens when I add less. Just know that it can tweak fluidity. Okay. The next thing that we want to look at is when we start looking at cell membranes, it turns out there's quite a bit of stuff in them. So back in the day, some scientists were trying to figure out what's going on with cell membranes. And they're like, hey, I need to see what's in there. So somebody was tasked with dissecting a cell membrane. So let's say these are the two parts. I've got the kind of the one side that was purple and the one side that was blue last time. Like this is the phospholipid bilayer, the bottom layer and the top layer. And they're like, I want to dissect the phospholipid bilayer. But don't dissect it this way. I want you to peel off the top layer. I want you to basically take the bilayer and sort of split it into like two monolayers. How thick is a phospholipid bilayer again? It's 1/9,000th the thickness of a sheet of paper. Is it made of oil? What happens if I had like, if I dumped some olive oil right here and I took a knife and I cut it? Does it stay cut? No, doesn't it just like seep back together? So how are you going to like get a phospholipid bilayer and somehow like split it that way and have it not go back together? So somebody's like, I know, how about we just like freeze it and then just like shake it really hard? And it worked. So they froze it, they shake it really hard, and when they do, like parts of it just kind of like break off. So sometimes, like maybe this very top layer right here just kind of breaks off. So I can literally see a monolayer underneath because the top layer of phospholipids, some of it just tore off. And when they looked in there, they're like, holy crap, there's a whole bunch of stuff stuck in there. So it's not just phospholipids. There's other stuff. And that's what you're seeing in this picture. If I zoom in on that bottom picture, that's an actual microscope image. This is microscope image. It's got some like, obviously, like generated art on the outside, but they just saw all these spots in there. There's another picture. This is where we take it and kind of rip it open. So that right there, you're looking at the phosphate heads from above. You're looking at like intact phospholipid bilayer from above. There's where it's torn. So now you're like looking through this little tear, and there's all these proteins sticking out at you. So they're like, hey, cell membranes have a whole bunch of crap in them. So now what we want to do is figure out what that crap does. So what does the crap do? All right, so think back to 9th grade. We had simple diffusion, or your teacher might have just called it diffusion. What was diffusion? Things move from like what to what in diffusion? Yeah. Yeah, high concentration to low concentration. So when I have a high concentration of stuff and a lower concentration somewhere else, things tend to move from high to low concentration. Now, I might remember that just because I've heard it so many times and that's why I remember it. What if I was sitting there and I was like, crap, was it high to low or low to high? Why is it logical that things move from high concentration to low? Why is it logical that like, oh yeah, I guess if I think about it, they wouldn't go from low to high? Yeah. Yeah, pressure's kind of built up. Things just kind of randomly move around until they're randomly spread out. So let me give you an example. Pretend I've got like 20 students over here. It's like a lockdown. And I suck, so I like put us right by the door. I don't know. So there's 20 of us over here. Right now there's a high concentration of students here. There's a concentration of like 0 students out here. And so we're all by the door because we're idiots. So now I'm like, all right, I'm going to let us go, and we're just going to all, like, it's almost like we're blindfolded, and we're just randomly moving around. So I'm just kind of randomly, you know, like I bump into something, and I bump into this. We just start randomly moving. If you come back in 10 minutes, aren't we likely to be pretty evenly spread throughout the room? Or is it likely that, like, all 20 of us are over here in this corner now? No. So didn't we start with a high concentration, and then didn't we spread out until we were at equilibrium? That's why things move from high to low, because they just do. Let me show you. If I've got a cup of water, there's a teeny tiny amount of oil in here, so we'll ignore that. And I got some blue food color. So just diffusion, you know that this is going to happen. It'll just start spreading out randomly. So eventually, it was a really high concentration in this little bitty blue ball at the top, and then it kind of hits, and then it's just going to slowly, like, spread around. And if we left it there long enough on its own, it would evenly spread as if I had kind of shaken it like that, right? Because they're just randomly moving. They aren't trying to go from high to low. They're just spreading out randomly. Got a fun trick for you. Okay, so I got some water here. I've got some oil. That's something you've seen before, of course. So here's some water. I have some vegetable oil, vintage vegetable oil. It's been sitting in the lab for I don't even know how long. That label's like old. I don't know how old this is, but I know I had a demonstration that I used in my 9th grade class this morning with some Surefine baking soda. Best if used by November 2nd, 2011. Now, I don't remember exactly when they built Timber Creek. It might have been 2010, maybe 2009, November 2011. So that's when, like, the due date was, which means that it was on the shelf in 2010, right? Where were you in 2010? Kindergarten? So this was likely purchased in kindergarten. You're like, I've never heard of Surefine. It probably hasn't existed since you were in kindergarten. So anyway, the vegetable oil is probably also pretty old. So based on what I just showed you, is this blue food coloring polar or non-polar? Polar, because it seemed to be happy to spread out in the water. What about the oil on top? Non-polar. So what's going to happen when I drop this blue drop onto here? There's oil on top, and then there's water underneath. Let me make the oil layer thicker. What do you think will happen when it touches the oil? I want to make sure I save some for next year. All right. So here we go. Let's see what happens. So we know all the blue spheres are polar because they're trying to touch the oil as little as possible. And the shape that has the lowest amount of surface area is a sphere. But then eventually, one of them touches the water and it bursts through. It's kind of pretty. So it's going from a high concentration, this little like blue sphere, and then as soon as it kind of breaks through, yeah, that would be a bad time for me to show. It's cool. I'm an athlete. So it'll like burst through, and they just sit there in the oil. I won't hold it over anybody's head. So that one busted through. We got about 10 of them on here. They're all just like floating around like perfect little spheres. So if you've already like watched all the episodes of your show and you're at home and you're bored, there you go. So get you through like 3 or 4 minutes. You got to find something else to do. Or maybe you just do this all night, I don't know. Maybe it's like those people that will just stare at lava lamps. So anyway, I can't leave this here, because otherwise you guys will just stare at it, so I got to hide it. Sorry. It's like that episode of The Office, if you watch The Office, where they're all staring at the DVD thing, and the DVD deal's like floating around. They're all waiting to see if it like perfectly hits the corner. You'll just be, I'll have this, be talking about all this biology stuff up there, and you'll just be like, that's for your own time. So sit there with a plate full of French fries and stare at that on your own time. All right, so simple diffusion. Simple diffusion, things move from a high concentration to a low concentration. You guys, regretfully, did not get to experience choir cookies. I think seniors didn't get that, right? You don't know what I'm talking about, do you? Okay, so the seniors that graduated last year might have been the last ones. You know how on Wednesday, how they batch up popcorn downstairs? You know what they did on Fridays? Cookies. So I think choir and maybe Sky Dancers or something like that did, they were called choir cookies, but I think other groups helped with them some. And they would batch those up fresh. By like second lunch, I'd open up my door, and the entire school just smelled like a bakery, just chocolatey goodness. And there were moms down there, like 6 of them, and they were just throwing those things out a dozen at a time. They were cooking so fast that one of the moms, her only job was to take a cookie, put it in like a little baggie, and put it on the stack. So starting at 8, she just did this for like 4 hours. And they couldn't keep them. You'd grab them, they're all warm. Oh, it's delicious. And then somebody's like, I don't think these cookies are healthy. And everybody else is like, no, done. They're like, I'm going to tell. And so, no more choir cookies. Thanks, Karen. You know? So, I don't know, that one's got walnuts in it. That's healthy, isn't it? It's like, not for that kid. He almost died. So anyway, simple diffusion. We're batching that stuff up downstairs. The smell just makes it all the way up to the second floor. It's not sneaking out my room. It just spreads out throughout the whole school. There are little bitty molecules of chocolate floating around, and when they hit a receptor in my nose, my brain goes, chocolate. So this is my cell. If I zoomed in on the cell membrane, I know that it's actually like phospholipid bilayer. So if I kind of like zoomed in on this black line right there, it's actually phospholipid bilayer. All right, cool. So over here, I'm going to draw a little bit of phospholipid bilayer. And I kind of start here because I end up showing you stuff all the way around to about here. So I end up drawing all over like this, like 60, 70% of the cell. So first off, number 1, simple diffusion. Again, your 9th grade teacher might have just called it diffusion, but in here we've got a few different types of diffusion, so now we have to distinguish between them. So, simple diffusion. I've got some phospholipid bilayer here. Again, every other tail is actually unsaturated. And so if I've got a whole bunch of these red dots outside the cell, and not very many inside the cell, then the red dots naturally can move from high to low inside the cell. Okay, that's easy enough. The next question. I'm going to have you talk with somebody nearby and see if you can figure this out. Okay, the first question is easy. You don't have to talk to anybody about this. What's more likely to be able to like slide through between these phospholipids? Because all these particles are doing is, just like we were seeing earlier, if this is my phospholipid bilayer, it's just like shuffling around. So aren't there like teeny tiny little gaps in there? If I was like trying to look at you, like a pirate or whatever. So there's teeny tiny little gaps as they're shuffling around. So obviously like smaller particles could squeeze through easier than large particles, right? The next question though, polar or nonpolar? So take 30 seconds. What do you think gets through the cell membrane easier? Polar things or nonpolar things? One has a much easier time and one struggles. Talk with somebody nearby if you want. Polar or nonpolar? Polar or non-polar? I know small things will have an easier time than larger things, of course. All right. So I can fully explain one of them, and the other one I can 50% explain. Okay, so how many of y'all think that polar things have an easier time getting through? Okay, how about non-polar? That's about a 50-50 split. Non-polar things get through easier. So the smaller I am and the more non-polar. So if you can't quite read that in this corner, it just says small plus non-polar. The smaller I am and the more non-polar I am, the easier time I have of getting through the cell membrane. Okay, so Mr. Cotton, why does it work that way? Well, if I've got my phospholipids like this, like that, outside here, is the environment surrounding the cell more polar or non-polar, do you think? Polar. Why? What's the main thing that's out here? Water. What about inside the cell? Water. So outside is polar, inside is polar. These lipid tails, what about those? Non-polar. Okay, so here's one that I understand. If you were to say to me, these green particles, they're polar. And I can tell you why they can't get in. Outside, it's polar. This molecule is perfectly happy hanging out here in this watery environment, right? It's hydrophilic. It's perfectly happy being right here next to these phosphate heads. That's cool. Does it make it through this barrier of lipidiness? No. So polar things basically just bounce back off. They're already in a place that they like. They don't travel across this like sea of non-polarness, the lipids. So that makes sense. I can explain that one. Here's the one that I can only 50% explain. Non-polar stuff. That just says NP for non-polar. Non-polar things. Is it stable out here in this watery environment? No. Is it very stable next to the phosphate heads? No. Is it happy to be in the lipid tails? Sure. So that makes sense. Here's the part that doesn't make sense. For some reason, it continues on. I don't know why. You're like, well, Mr. Cotton, how long have you not known why? About 15 years, 20 years. You're like, haven't you ever looked it up? Yeah. You got that cell biology book. Don't they explain it? Yeah. You got another cell biology book back there. I can go online, and there are cell biology books, and they all say the same thing. They'll be like, this is a non-polar thing. It's not stable out here. It slides through. It's perfectly happy in the tails. And then it keeps on going. And I'm like, wait. What do you mean it keeps on going? And they're like, and it keeps on going. They don't say. Sometimes in science, they'll be like, we understand this, and then we don't understand this. That makes sense. When you're just like, hey, we know this happens, but we can't explain why. Why is it that phospholipid tails are almost always even number of carbons? We don't know. But phospholipids, the tails are almost always 12, 14, 16, or 18 carbons long. They're like never odd numbers. We don't know why. But if they say we don't know why, I can accept that. So, I've been saying this for as long as you guys have been alive. If any of you guys go off to college and you're in some cell biology class and some professor explains why that non-polar thing keeps going, send me an email. Let me know why. It's probably there's some protein that pulls it through. But every book just glosses over there like they don't say a protein probably pulls it through. They're like, and it keeps on going. And I'm like, that doesn't make any freaking sense. And I get mad. So if you guys figure it out one day, send me an email. I'd like to know. All right, so that's simple diffusion. Okay, do you know a word, an F word? Hang with me. An F word that means to help. To what? Yeah, facilitate. So the next type of diffusion is facilitated diffusion. So we'll do that one right here, number 2. Facilitated diffusion. Facilitate, that's a C. That's an L. I can't remember if it has 1 L or 2s. I always want to write 2 L's, and then when I write 2 L's, it looks wrong. So I'm sure I've got it in your notes there somewhere. But facilitate, it's either got 1 or 2 L's. I don't know which. Facilitated diffusion. Again, facilitate means help. As a teacher, if I'm a good teacher, then I facilitate learning. If you had an hour to learn something on your own versus an hour with me, if you're with me, if I'm a good teacher, then like you learn more stuff, or it's more efficient, or you have fewer errors. If I facilitate learning, you're better off with me teaching you biology than just learning on your own. So facilitated diffusion. Facilitated diffusion, there's a few different types. One type is I'll use a protein channel. And they usually draw protein channels like that. So one type is using a channel. Sometimes they call them pores because they act like little pores. Stuff can leak in and leak out. But usually they call them channels. So that's a channel. Why would I need that? Well, if something's small and nonpolar, I don't. So small nonpolar things, they just slide between the phospholipids. But let's say I have something that's polar that I need, like water. Now this polar thing has a way to get through. It gets rejected, but if I have a channel made for it, it gets through. This is how they draw channels. However, it's a little bit of a lie. So I'm going to show you how I think they should draw channels. Again, you can draw it like this, but I think of it more like this. It's almost like a maze. This channel right here makes it look like anything smaller than like the size of a dime could just slide through. If I had a chemical that's, as long as that chemical is about that big, no matter what it is, it could slide through. Most channels aren't like that. Most channels are specific for like 1 or 2 things. So I'll have a channel that only lets water in or out. How does it only let water in or out? All these little nooks and crannies of amino acids in here, they all attract water. So like that part attracts water. Then the next part attracts it harder. The next part harder, 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 until the water goes like all the way through. And other molecules have a difficult time getting through. So the best analogy I've got is this. It's dumb, but you'll still probably remember it because it's dumb. Pretend that I've got a doorway and there's a cutout, but it's exactly the shape of me. Like, I have to walk through like this and like turn my foot sideways, and it's an exact cutout of me. So I'm about 5'11", so it'd be 5'11", but it has to have like the exact same dimensions for my hand, the length of my foot, the width of my foot, the width of my shoulders, waist, like every single thing fits down to like 1 millimeter. So are there other people that are 5'11" in this school? Sure, that's a very common height in this school. But do they also have the same size hand, the same size foot, the same body frame, the same, like at some point, there might not be another person in this school that has the exact same outline as me. So this particular thing, I'm the only one that can walk through there. And I have to walk through like this. That's what the channels are like. There's one, for example. Called aquaporin. It ends in IN. So what can I assume about it if it ends in IN or EN? It's a protein. It acts as a pore, and what does it let in? Yeah, aqua, water. So it's a protein pore, a protein channel, that's specific for just water. I've got one for sodium. I've got one for potassium. I've got one for chlorine. I've got one for calcium. So all these things that I have channels look like when we draw them. It looks like just any old thing that's small enough fits through there, kind of like this doorway. Every student can fit through this doorway. Unless I'm like a 5,000-pound man and I'm like, you know, like that or whatever, then I can fit through the doorway. But, you know, otherwise, like in reality, they're all very, very specific. It's all right. It's a fictitious person. He doesn't care. So facilitated diffusion. It's helped by a protein. Did I require any proteins to help me over here? No, so that's just simple diffusion. Here, can this polar thing get through without the help of a protein? No, so it needs help. Facilitated means helped, so it's helped diffusion. All right. Somebody decided that if water is going through a water channel, that it should get its own special name. We don't just call it facilitated diffusion of water. What do we call it when water moves from high to low? Osmosis. Why does it get its own name? I don't know. When water is going through a channel. If you were to Google it, the true definition is osmosis isn't just when water is moving, but it has to be going through a channel. Has to be going through a pore. It has to be helped by a protein. But instead of calling it facilitated diffusion, they call it osmosis. I don't know why. You're like, all right, that's fine. That's fine. You heard about channels back in 9th grade. You may or may not remember it, but you did. Now we're going to get into some of the ones that it kind of depends on the teacher that you had. One of the problems with channels is they're just wide open. So if I've got like 80 of these green particles here and 30 here, they're going to always be moving in. If it's water, fine. If it's glucose, fine. Glucose automatically moves in, and as I use it, more diffuses in. As I use it, more diffuses in. So I always have glucose. I always have an energy supply. So that could be good. However, what if I've got a bunch of these red dots? So here's number 4. What if I have a bunch of these red dots, and I want to let them in? I want more. I mean, the red dots. Of course, everybody wants more red dots. There's only a couple of red dots inside. I've got a bunch of red dots outside. If I have a channel, they're just going to move in whenever they want, and I don't have any control. So what can I do to make it to where they can only move in when I want them to? Yeah, I close it. I put a gate on there. So now it's got a gate on the channel. What would be a good name for that? Gated channel. So this is a gated channel. I can open and close it when I want. So if I didn't want my students thinking about choir cookies quite yet, let's say it's 3rd period and the smell of chocolate is strong, I can leave the door shut. And then like it's time to turn on TCTV and I want to like release the aroma of choir cookies. You can tell I miss these. Because it reminds you, you open the door, not only was it like it's choir cookies, but it's also basically telling you it's like Friday. And you're like, yeah, it's Friday, choir cookies. You know what I mean? It's like, I don't know. It was so nice. So you open this up, and now the choir cookie smell diffuses in. All right, so I can open and close this channel at will. We study this more later, but I want to try to convince you that you need to remember this and that it's interesting. So anytime that we're learning something important, I either try to have a funny story, an interesting disease, or just something interesting about it. So we study this more later again. This will be about a 3-minute little side tangent. How many of y'all have finished anatomy and physiology? Maybe you took it last year. 2. How many of y'all are in it now? 3. Okay, so if you finished it last year, then you studied the nervous system. If you're in it right now, you're getting ready to do skeletal, and you haven't done the nervous system yet. So only a couple people. Have any of y'all finished psychology? Okay, so people that finished anatomy and finished psych might already know this. Everybody else, you might not. So this is a nerve cell, like a brain cell. We think that the average human brain has up to 100 billion nerve cells, 100 billion neurons. Some studies say as low as 80 billion, some say as high as 100 billion, so I'll say tens of billions. And this is basically like what my brain looks like on a cellular level. I just have like tens of billions of nerve cells. That's a nerve cell. That's one. That's one. That's one. That's one. So I've got like 25 nerve cells up here. Okay, so play along with me. I know this is an easy question. What is 6 times 6? 36. So somewhere in your brain, a bunch of nerve cells lit up with electricity. So maybe when nerve cell 1, 2, 3, 4, 5, 6, 7, 8, when electricity flies through here and then it's transferred to here, and then flies through nerve cell number 2, and then it's transferred to nerve cell number 3, 4, 5, 6, 7, 8. That's where you store the memory of 6 times 6 is 36. All right? Play along with me again. What's 6 times 5? 30. That's a different pathway. That might be 1, 2, and I'll just kind of continue the numbers, 9, 10, 11. And that's where you store that memory. If I say, imagine the color red, this part of your brain lights up. If I said, translate red into Spanish, and you're like, rojo, and you're like, man, you sound like a native speaker. All of that is right there in like different nerve cells like firing in my brain. Can you choose to quit thinking about 6 times 6? All right, what's 6 times 6? 36. If I say now, what's 7 times 5? What's that? 35. So you decided to quit thinking about 6 times 6 and think about something more important like 7 times 5, right? And then if I say, what's Spanish for blue? What's Spanish for blue? Azul. So you chose to quit thinking about numbers and think about Spanish. So you can choose when to let these nerve cells fire and when not to. That's because when you're having a thought, these gates are opening and closing. When they're open, you're having thoughts. When they're closed, you're not. We study the nervous system more in the future. So if you're like, I don't know that I get that, that's fine. I'm giving you like a 3-minute summary of how nerve cells work. I'm summarizing like a 90-minute class. But basically, anytime you have a thought, learn a skill, access a memory, move your arm, anytime your brain is telling your body to do anything or you're mentally thinking of something, you just have like these sodium channels that are gated, potassium channels that are gated, and then a sodium-potassium pump. Sodium channel with a gate, potassium channel with a gate, sodium-potassium pump. Right now, that nerve cell is not being used because I'm not thinking of 8 times 8. Oh, wait, now I am. So that opens, and a bunch of sodium and potassium starts moving in and out, and that creates electricity, and that is a thought. If I ask you, next time I see you guys is Tuesday. If I ask you on Tuesday, hey, what was that F word that meant help? If you remember, what happened was you found that pathway. Maybe 1, 2, 3, 4, 5, 6 is where you store facilitated. So I'm like, hey, what was that F word that meant help? This part lights up, and you go, facilitated. If you forgot it, I'm like, hey, what's that F word for help? And you're like, freaking, I don't remember. I don't know. And that is your brain can't find the pathway. That's forgetting. How do I decide to think of 4 plus 4 versus my favorite movie, or I can picture this person, or I can think of my favorite song in my head? I'm just opening up channels in certain nerve cells, and if the right ones open up, I think that thought. My brother is 54, and he's been playing guitar since he was about 14. He's incredible. He's probably averaged 25, 30 hours a week of playing guitar ever since that time. So he can just sit there and just play whatever. He can listen to some like 80s guitar solo and like figure it out and just like play it. Like he'll play some Van Halen, whatever, some Led Zeppelin thing or whatever. Give him like 5 minutes and he'll be like, oh yeah, I can play that. And then he can sit there and talk to you while he's like playing this really hard song. He'll tell me like, play a G. And I'm like, I don't play guitar. And I'm like, he's like, hold your hands like that. And I'm like, Bring. I'm like, man, you got to push hard on those. Bring. Bring. Oh, yeah, that's G. You guys in music, you know, spot on G right there. I have no idea. And then I go to, like, I don't know, B. Every good boy does fine. I think these are all legit. So then I go to this, and I'm like, What I'm doing is my brain is trying to figure out all the nerve cells that have to fire in just the right way to make all my muscles contract in just the right way. And then I have to go back to G, and so I have to find that perfect pathway again. I'm like, dang it. And then eventually, if I do it like 10,000 times, I'm just like G, 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 G to F, G to F, G to F. 4 times 4 is 16. 5 times 5 is 25. 8 times 8 is 64. What's 17 times 24? I don't know. I haven't rehearsed that. Now I've got to sit down and like do that math in my head. But what's 5 times 5? 25. What's 3 times 3? 9. I've rehearsed those so many times. 918-258-9838. That's my childhood phone number. Haven't lived there since I was 13. That's just buried in my head somewhere. So gated channels mean I can choose when to do something and when not to, kind of like thoughts. T-H-O-U-G-H-T, thoughts. All right, that's a gated channel. So far, everything over here is all passive. If it ever has diffusion in the name or osmosis in the name, it has to be passive. What do I mean by passive transport? What does it not need? Energy. So everything this way is all passive. All passive transport. Now, one thing that might bother you is you're like, wait, doesn't your body have to burn energy to open that gate? It depends. However, once I open the gate, once I open the door, doesn't the popcorn smell on Wednesdays just kind of like permeate through here on its own? I don't have to grab it, it just does it on its own. So scientists have decided, even if it does, make me burn an ATP to open that gate. Usually it doesn't, but even if it does, then the stuff still moves in on its own afterwards. So they still decided to classify it as passive, even though you don't like it. And that means the cell isn't burning energy. Of course, when things are moving in, that's kinetic energy. Like the cell doesn't have to do anything. I just open the door and the popcorn smell just drifts in on its own. I don't have to like grab the popcorn smell and like throw it at you guys or something like that. All right, so that's passive transport. What's going to be on this side? Active transport. So now, let's look at active transport. You likely learned one type of active transport protein in 9th grade, and it even kind of sounds like it would be active transport based on the name. Does anybody know what that kind of protein is called? It does start with a P. It's a short word. It's called a pump. So when I draw pumps, I usually just make them look like rectangles. Pump. So number 5 here is a pump. Pumps are very specific, just like these channels. Like I said before, I might have a channel that only lets sodium in, and I'd call it a sodium ion channel. I might have one that only lets potassium move. That would be a potassium ion channel. So these pumps might be like a sodium pump or a potassium pump. There's even a sodium-potassium pump. It moves both. So why would I ever use a pump when I could use a channel? Like, let's say this is a sodium pump, and that requires energy, which means I have to eat food. Wouldn't it be easier to just like have a sodium channel and then like I don't have to eat food? Do what? Yeah, maybe the cell needs it inside. Okay, what if I've got, let's say that I've got a bunch of these dots inside, the purple dots. I've got like however many that is, 20, 25, something like that. And there's only 2 more purple dots outside. But I love purple dots. So if I had a channel, if this wasn't a pump, if it was a channel, which way would the purple dots move, in or out? Out. That's the opposite of what I want. So passive moves from high to low. Which way does stuff move with active transport? Low to high. So if I want to force something against nature and go from low to high, low concentration. That's the chemistry abbreviation for concentration. Low concentration to high concentration. Then I need active transport. And the simplest version of that is a pump. And then we name the pump based off of what it does. If it's a proton pump, it pumps protons, and so forth. So pumps. This is why we have to eat food. This is not the only reason, but this is a reason we have to eat food. Because sometimes I want more purple dots, and I'm stockpiling. And that takes energy, so I have to eat food, convert it into ATP to run this pump. Active transport needs energy, and usually, not always, usually that energy is ATP. Usually. We'll learn some exceptions as time goes on, but usually ATP. All right. So now, here's something that's kind of clever. They consider this to be active transport. I don't think it should be called active, but books classify it as active. Okay. Let's see if we can figure this one out. I'll try to set up like a logic puzzle for you. All right. What if I want some purple dots, just like here. I want them on the inside. So I want those purple dots to move in. Does that require energy? To go from here to here? Yes, because I'm going from low to high concentration. So that's going to require energy. Okay, what if I don't want to burn an ATP? However, what if nearby, are any of y'all red-green colorblind? You are? Okay. Purple versus green, can you tell? Or does purple have too much red in it? It is? Okay. 1 in 30 males are red-green colorblind. It's a sex-linked trait, so it's way more common in males. We should statistically have 2 females in all of Timber Creek that are red-green colorblind. But every other classroom should have a colorblind male, because every 30 males. So average class is 30. Average class is like 50-50 males and females. So about every other class should have a colorblind male. But like the senior class in its entirety might not have a colorblind female. So, all right. So let's say that I've got these green dots also. And I don't give a crap about green dots. I mean, who cares about green dots? I don't care what happens to those. If they move in, fine. If they move out, fine. All right. Do you think there's any way for the green dots to help power the movement of the purple dots? Again, I don't care what the green dots do. I want to pull in those purple dots. Let me give you an example. Let's say that you go to an airport or a fancy hotel or this one restaurant in South Lake, and I don't remember what it's called, rockfish, something, I don't know, they sell fish. You ever seen those like rotating doors, the circular doors? Nowadays, they're like powered. So back in the day, you at least had to push them. Nowadays, they're like, oh, we make people push doors. Maybe it's, you know, anyway. So now you like walk through and it like rotates like this. What if, pretend I have this little baby. This is a weird analogy, but it works. All right, so what if I have this little baby and he's already loaded in the door. So this door from above, it looks kind of like a pie, right? Like from above, it would look like, you know, like a bird's eye view. I could be in this one, somebody else could be in that one, and then like I'm pushing it or whatever. So let's say the baby's already loaded up in there. You're like, did he just describe this as loading up a baby? Yeah, I did. So there's this baby, and he's right here. He has no energy. He can't push the door because he's a baby. There he is. We want the baby to enter. We want the purple to enter. But he has no energy. What if I'm behind him, though, in the section behind him? What if I push? Now doesn't that sort of like force him in against the concentration gradient? So what if the energy from green moving in, diffusing in from high to low, is able to like push the purple in? It does. That's a thing. They call it co-transport. Co-transport. Some books, this is what's annoying, in AP Bio, there is no official AP Bio book. There's about a dozen. So like, you guys are using the book by Hillis. That's a professor at UT Austin, by the way. Your book is written, there's like 5 authors in it, and the main author is a professor at UT Austin. Down the street at Northwest, they might be using the book by Raven. In Southlake, they might be using the one by Campbell. In fact, I used to teach in Campbell, and they use Raven. We're all taking the same test in May, and we aren't even using the same book. So some books call it co-transport. Some call it SYM, S-Y-M. Now, I don't care if you misspell it, but the spelling is important here, S-Y-M. If when you were writing it, you spelled it S-I-M, we'd give you credit. Why do you think this is called co-transport or SYM transport? SYM, think of geometry. Which way are the purple ones moving? In. Which way are the green ones moving? In. Aren't they going the same way? SYM. Like symmetry. They're the same way. Co. They're going together. There's another one that does the opposite. Now, let's say that I want to get those purple babies to enter. I want those to enter the room. But now, this time, the green ones are heavy on the inside. And there's not that many outside. So now, which way will the green ones move? In or out? They'll move out, because the green ones are going to move from high to low. They're just doing regular old diffusion. So they're going out. And we're wanting to pull in the purple babies this way. So now they're going opposite ways. So we don't call it co-transport, we call it counter-transport. Some books call it anti-transport, or they'll call them like anti-porters. Anti-transport, because anti means opposite. Counter means opposite. Somebody attacks, so I counterattack. If I'm against transport, I would be anti-transport. Like, I'm against transport. Make a little sign. I'm against transport. So now, in this analogy, it would work like this. Now let's say I'm the green dots. So I've got a bunch of green dots. I've got all my green expo pins. Sorry if you're watching the recording, this is not going to come through. Got the purple dot, baby. He's loaded up in the thing. I'm green dots, so I naturally move out. And as I do, that just sort of like shoves the baby in, right? So I'm kind of like, as I push out, that's like forcing him in. Yeah, I don't know where it ended up. If you find it, let me know. Oh. Oh, it's probably trapped under your bag. Oh, there it is. Thank you very much. So that's counter-transport. Same idea. Diffusion one way powers the movement of something else. To me, overall, that seems like I'm energy even. It seems like the cell's not really using any energy at all, but all books call this active transport. I would have called it passive because, again, it's almost like I got plus $5 and I spent $5, so I'm still at like 0. But books call it active transport, so, you know, whatever. All right. Let's look at some interesting applications of some of this stuff. Let's say, something interesting we got here. There's some kidney stones. There you go. Look at this picture here. And for the video, switch cameras. Look at this picture. This is a beta barrel. It's made of beta pleated sheets, and it looks like a barrel. Have any of y'all ever had strep throat? Yeah. So you go to the doctor, and you're like, my throat hurts. And your doctor, mine happens to be a male, so I'll say he looks at my throat, and he's like, I think you have strep. And he's like, and I'm like, why? And he goes, it's like a mess back there. Everything's all swollen. So we're going to get a sample and see if that's strep bacteria or not. Streptococcus pneumoniae, I think, is the one that causes it. Maybe a different one. But they're going to go in there, and they're going to take that big old long throat culture thing, and you're like, and they like, there's literally like bacteria growing in the back of my throat. So they're going to scrape some off, and they'll be like, all right, wait here for like 15 minutes. They give it to some dude back in a lab. He smears it on a microscope slide. If any of y'all have taken microbiology, he'll gram stain it. He'll come back, look at it on a microscope slide, and he's like, yeah, that's strep. They'll be like, you tested positive for strep. That test costs $80. And you're like, didn't that guy gram stain that slide and look at it in like 4 minutes? They're like, yeah, it's $80. And you're like, okay. So I've got strep. Well, how does strep work? Turns out strep and some other things do something similar, is they build this protein with this beta barrel. And that beta barrel makes just a big old pore. Like that. And what it does is it inserts in my cell. So what happens if all of a sudden I just start poking a whole bunch of holes in my cells? Like, does stuff just start moving in and out at random? Do I have the right balance of sodium and purple dots and all? No, purple babies are like spilling out everywhere. Like there's all kinds of crap going on. So water just rushes in, water rushes out, all kinds of stuff, and my cells die. And that's why my throat hurts. Because all my cells in my throat are dying. Because they've got holes in them. Because of strep. Who would have thought? So they build channels. It's kind of crazy. All right. Now, if we start looking at like a cell membrane, we might have channels. This one's always open, like these. Might have a gated channel, like this. Transport protein, I try to stay away from that word. Technically, transport proteins can grab stuff and passively move it, but I'm going to avoid that one. I can have pumps in there. I can have other stuff. So the next one that we'll look at is bulk transport. Or just for the fun of it real quick, have you guys ever seen kidney stones? Yeah. You're like, why are they red? Why do you think they're red? Yeah. So in my kidneys, number one up there, our kidneys. Your kidneys are like, you'd have to move your intestines and everything out of their way. They're like embedded in the back muscle of your wall. If I get kidney stones in there, I have to pass them down this tube, which goes to my bladder. And then I have to pass it from my bladder out my urethra. That's your P tube. Unfortunately, though, they're shaped like that. They're not shaped like BBs, nice and round. So what are they doing is they pass through all these tubes. They're just like scraping it along. So why are they red? Because of the blood. Their natural color is not red. Their natural color is this. When you pee them out, they're red. Now, I've never had a baby. I'm a father, but I've never birthed a baby. Women that have passed kidney stones and also had kids through natural childbirth say that they're comparable. I've never passed a kidney stone for either one, but women that have done both, many women, not just like one rando over here or whatever, but many women say like, yeah, passing a kidney stone is about like birth. You know, also depends on how long it takes and all that. But it's supposed to be, you know, painful. Anyway, so hopefully you never have to do that. Do any of y'all have close bloodline family members? Not like a stepbrother, because that's not blood. But do any of y'all have like brother, sister, bloodline father, mother, or grandparent that had kidney stones? The best predictor of whether or not you're going to have kidney stones is, did you have a close blood relative that did? The best predictor of whether or not you're going to have a second kidney stone is, have you ever had a first? If you're like, my grandfather's passed like 4 kidney stones, and my dad's passed like 3, you're going to be pushing out kidney stones. Is there anything I can eat or drink differently? Not a whole lot. There are some things that in theory could make it a little bit worse, but overwhelming, the best predictor is genetics. Just like your eye color or hair color, it's not what mom or dad consumed. The main predictor is like genetics. So it's not, well, my mom had Pepsi and that's why I've got blue eyes or something like that. So anyway. All right. The next one that we want to look at is bulk transport. So far, everything that I've looked at, I'm moving things like 1 or 2 or 3 molecules at a time. So this is still active, but it's going to be bulk. These are also ones that you've learned about before, and you'll find these to be pretty interesting. So bulk transport, just like buying in bulk at Sam's. So here's a picture of one. This, actually, what do you guys see in this? I'll tell you what it really is, but what do you see? A what? A gun? I see a bird with a big old, yeah, like a parrot or a toucan. It's standing like this. These are its wings at its side, and it's got like a big old bird beak. And it's like on a tree branch or something. So anyway, what it really is, it's a digital image. So if you use an electron microscope, they're digital images. So just like anything digital, you could make it whatever color you want. So these are not the natural colors. But that orange stuff, that's E. coli bacteria. And the yellow is a white blood cell gobbling up E. coli bacteria. So this is a type of bulk transport. So we can kind of break it down into like a couple of categories. Endocytosis. We're going to learn 3 types of endocytosis. Might not get to all of them today, but we'll learn 3 of them. Endocytosis. If you've had like medical terminology, or maybe you're in like the HOSA type of pathway through KCAL, what does osis mean? Like in a midterm class, you might have to know what certain things mean. Like, what does pneumo mean? Or what does itis mean? What about osis? It's funny when like everybody turns around and looks at like 1 or 2 students. They'll be like, osis means a state of or a condition of. That's why a lot of diseases are in osis. Osteoporosis is a condition of porous, what's osteo? Bones. So it's a condition of porous bones. Scoliosis. So when I look at endocytosis, what is CYT? Cell. And then what is endo? Inside, entering. You might have heard of like endothermic reactions in chemistry or physics and exothermic reactions. Endothermic, I'm absorbing energy from the environment. Exothermic, I'm releasing energy to the environment. So endocytosis is a condition where the cell is bringing stuff in. Well, one of those is called phagocytosis. I'm a high school teacher, so I say phago. Phagocytosis is cell eating. So if I am digesting something that's big and mostly solid, they call that phagocytosis. So when a white blood cell gobbles up bacteria, that's phagocytosis. Pino. If phago is cell eating, if you had to guess, what's pino? cell drinking. So if I'm bringing in mostly solid stuff, I call that eating. If I'm bringing in mostly liquid stuff, I call that drinking. Just like at lunch, I don't eat my water, I drink it. I eat my pizza, I don't drink it. So phagocytosis, if I'm bringing in mostly big solid things, I call it phagocytosis. If I'm bringing in mostly liquid stuff, I call it pinocytosis. So some of these pictures always kind of fascinate me that all this crap is going on. The thing that I love about this class is most of the time, whatever we're talking about is like happening in my cells right now. Like all this crap is happening right now. Like things are shuttling around. Like as I was telling you about gated sodium channels and gated potassium channels, and that's how my brain works. That's literally what my brain was doing. So my brain was talking about how my brain was working. So right here, I've got a cell. The cell is like, you know, up here, like that. Let me click over for you. My cell is like right here, and this part just kind of like pinches in. And there, I don't really see like a giant thing, so that's basically cell drinking. Here, when the cell pinches in, that's bringing in like E. coli, so that's like cell eating. So what it looks like is this. If this is my membrane, it just sort of like pinches in. Here we go. So pretend I've got this E. coli out here. He's sad because he's getting ready to get killed. He's untrustworthy, so let's give him like a faint mustache. All right, so what will happen is the cell just kind of like pinches inward. The whole phospholipid bilayer just pinches inward. It's like blowing a bubblegum bubble in reverse. Some of you guys have done this. So normally you blow a bubble, and whether you realize it or not, you're taking the gum, you like form it into a sheet behind your teeth, you stick your tongue through it, and then you like blow air through it. Well, now to do a reverse bubble, you form it into a sheet, you put it on the front of your teeth, and you suck in. You guys know what I'm talking about? It's a reverse bubble. If you ever wonder what your parents did before the internet, crap like this. Hey, you know what I learned how to do? I learned how to gleek. Do you know what gleeking is? It's where, yeah, if you don't, there's spit where you're like, and that's all fizzy and gross. So it's got air in it. Gleek is where you like pull your tongue back, you somehow smash salivary glands, and this perfect stream, no bubbles, of saliva comes out. What'd you do for entertainment during 3rd grade, Mr. Cotton? I tried to learn how to gleek. I didn't succeed. What about 4th grade? I learned to do this. Success. Can't do the other side. That was 5th grade. That year was a wash. So, a reverse bubble. So now, this thing basically ends up inside. And then we'll learn just a little bit later on in this unit that lysosomes will come and like destroy him. So a white blood cell will sort of gobble up and then sort of like digest this bacterium. All right, what if I've got all this dissolved crap just outside my cells? Let me flip on this slide. What if I've got all this stuff dissolved in liquid on the outside of my cells? And again, we're Timber Creek, so we like the purple dots. It's a school color. There you go. I remember I was in this one class, it was an English class. We had to like write something and put it on paper, and then we're supposed to decorate it. So to decorate it, I got like 8 different markers, and I pulled the caps off, and I went. And she's like, what is that? And I was like, confetti. She's like, I don't think that's very good. So let's say I've got these green dots, red dots, purple, blue, whatever, but all I want are the purple dots. Here's the problem. I could have a channel, and it brings in purple dots one at a time. I could have a pump, and I'm bringing in purple dots against the gradient, 1 or 2 at a time. This is super accurate, but it's slow. It's kind of like whenever we first started the whole show your badge when you walk in the North or the South Hall in the morning. We just had one admin. There'd be this long line. But if we looked at it, you can go. That'd be accurate but slow. So pinocytosis, bulk transport, is when I want to go faster. But sometimes I sacrifice accuracy. If I've got a math assignment that's due tomorrow, pretend that tomorrow is not Saturday, it's due tomorrow, and if I can get 100 on it, I get an A in the class, and I don't have to go to class anymore this year. I'm going to do that homework assignment, and I'm going to look at it, revise it, check it, look on the internet, have my friend look at it, like that thing is going to be accurate. If the homework assignment didn't even count, I might like rush through it, and I'd be like, oh, no, I think the answer's 7. I want to go hang out with my friends. And I'll be, a lot less accurate. I'll be faster. So why do I have sodium channels, active sodium pumps, but I can also bring in sodium this way? Do you want speed or do you want accuracy? You don't get both. So with pinocytosis, I just pinch inward, and as I do, I'm now going to get all this crap out here. So am I going to get tons and tons of purple dots all at once? Yeah, but aren't I also getting green dots and red dots and blue dots and whatever? Yeah. So it's fast, but not very accurate. Over here, one at a time. Slow but accurate. So what do you want? So my cell has to decide, do we want accuracy or do we want speed? The last one we'll look at is my favorite one. I'm sure it'll be your favorite type of transport also. I think I've given you guys hints of like good material you could bring up on dates, right? Things you could talk about. You talk about like your baseline body temperature. Was that this class? So I got another one for you. After you use the baseline body temperature, I recommend date number 4 for that one. Date number 5, you're like, crap, what are we going to talk about? What's your favorite type of endocytosis? Now, the only bad thing is everybody's favorite type is, right, receptor-mediated endocytosis. So it's kind of an easy one because everybody always says the same thing. So RME, receptor-mediated. If that word's fine, I don't know that it's one I would have picked. Receptor-mediated, and then this is a type of endocytosis. RME, receptor-mediated endocytosis. Mediated, like mediate with a D, like I'm a moderator, mediator, whatever. Similar sounding words anyway. So here's how receptor-mediated works. These things, pumps, channels, really accurate, but really slow. These things, phagocytosis, pinocytosis, very fast, not very accurate. It'd be nice to have like a medium. Is there a medium speed, medium accuracy? That's receptor-mediated endocytosis. So here's what will happen. Let's say I've got a bunch of crap out here. So I've got all these dots that I don't care about. But among those dots, I have these purple triangles. And purple triangles are pretty awesome. So I'd like some more of those. This is kind of the unit where some students start showing up as the year goes on. This is about the time that I start noticing like people get ready for class and they pull out like 3 or 4 different colors of pens and pencils and stuff. You don't have to, but some stuff, it helps. All right, I want the purple triangles. I don't want to do this because that's too inaccurate. I don't want to pull them in one at a time because that's too slow. I want to be medium speed, medium accuracy. So what my cell will do is it'll manufacture receptors that fit purple triangles. And it sends them to the top, to the front of the cell, up here to the membrane. So now, those purple triangles, if they bump into those receptors, they'll stick. And then if I pinch inward, I'm more likely to get at least 1, 2, 3, 4 triangles. So think of it like this. Pretend that there is dust in the air, like actual, like, I don't know, it's like road construction type of dust in here. There's dust in the air, and I want to grab a bunch of dust. But whenever I try to grab dust, I'm going to also be grabbing like carbon dioxide and nitrogen and oxygen and all the other components of the air. But I want just a bunch of dust. So what I could do is I could get like some sort of sticky shirt with like tape all over it. So I've got a shirt, I got a bodysuit of tape. Probably exists. I've got a bodysuit of tape, and I just stand here while the dust swirls around. And I stand here for like 10 seconds, and then I pinch inward. Aren't I going to have a good amount of dust sticking to me? Yeah, the receptors are kind of like the sticky tape. Whatever's circulating around out there that I want should be able to stick in there. We can see this in these pictures. This is one of the things, specific things that fascinates me about biology is this is how it works. There's all these proteins involved. Kind of pinches inward like this. And the next picture, this will be the last set of pictures that we see, is going to show these proteins like that are kind of helping pull this in. You're like, hey, does it actually look like that? That almost looks like it's forming like a soccer ball. Like if I was inside of a cell and I looked out and I'm doing this receptor-mediated endocytosis, does it actually look like that? Well, these are the proteins. They look kind of like Ys. I drew mine as Ys. And if I take all those Ys and pull them in, they make like little bitty soccer balls. This is an actual microscope image from the inside of a cell. So let's say that our room is a cell. And right now, this wall is doing RME, receptor-mediated endocytosis. It's pinching inward. Why? 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 Why am I still alive? I can't believe that this works. And so this is like pinching inward, and it's got this bubble of like the purple triangles that I want. And it's doing it all the time. On average, 2,500 times every minute in every cell. So my body is just searching for specific cargo and grabbing it. It's almost like, here's tape that will attract popcorn smell. Here's tape that will attract pizza smell, whatever, and I just grab whatever I need and like pull it inside the cell. Receptor-mediated endocytosis. All right, the bell's going to ring in like a minute or less, so see you guys later. Stop recording.